Hello, my name is Aaron Opperman, student in BIO202, and this is my presentation on the influenza virus. Influenza virus is actually 3 different strains of the same virus. They all come from the same family, Orthoviroidae. Types A and B have a higher morbidity rate and can be more serious. Type C manifests as a less severe respiratory infection, mainly in younger children. The influenza virus is a spherical and covered in a lipoprotein envelope. The outside of the envelope has glycoprotein spikes, used for recognition and attachment to the host. Ion channels to regulate the internal cellular fluid are also present. Type A and B have 8 strands of single-sided RNA strand, and type C has 7 RNA strands. Transmission of influenza is usually by direct contact with the infected individual, or by aerosolized droplets, such as sneezing or coughing on somebody. It can also be transmitted by formites on the surfaces and objects, but this is less frequent. Poor ventilation can add to the transmission. These can include tight quarters such as classrooms, airports, airplanes, large gatherings, and military installations. The drier the season, this also contributes to the spread of influenza by keeping contaminants airborne for longer. Also, microfissures in the respiratory tract allow the virus to more easily enter the host. You can see this demonstrated very well here by the infected individual, the susceptible individual, by droplet, direct, or indirect contact. The influenza virus causes influenza infection, or commonly called the flu. The incubation period is around 1 to 4 days, depending on the type of influenza. The signs and symptoms also vary widely within the type of influenza, the year of infection, and what mutations are seen histologically. In general, chills, fatigue, and lethargy are the most common signs. Most influenza presents with a high fever, a dry cough, and a runny nose. Type A and B can also lead to death in some untreated cases. The influenza virus is a highly infectious and can have a high morbidity rate if the environment and the host are correct. 2 main glycoproteins on the influenza virus allows it to attack the human host. Hemagglutinin, named for its ability to agglutinating erythrocytes, and neuraminidase, which breaks down the mucus lining in the respiratory tract. The virulence factors mainly come from the antigenetic drift and antigenetic shift. Antigenetic drift is the ability of the influenza virus to mutate certain parts of its envelope, and not others. The antibody binding of the external glycoproteins that have a high rate of mutation. This allows them to become more resilient to the human defenses. However, the anchoring sites on the same glycoproteins have a low rate of mutation, meaning that they still bind to the mucus layer of the respiratory tract. Antigenetic shift, on the other hand, is when different viral strains exchange RNA copies when infecting the same host. This can lead to further mutations and adaptations of the virus. When someone says it's H1N1 virus, they are naming the new mutations of these 2 glycoproteins. The influenza virus is endocytosed into the respiratory cells, and RNA translation happens in the nucleus. Budding then releases the formed virus and allows for further infection. The glycoprotein neuraminidase is able to bind to the ciliated respiratory epithelium, which causes it to shed the layer of mucus protecting the respiratory tract. This obviously leads to further viral replication and the symptoms of a dry cough. Second, The immunological response of inflammation in the lungs is termed the cytokine storm. Counterintuitively, the influenza virus can be more harmful to healthy individuals because they're able to produce a higher immune response with little effect on. This is due to the antigenetic drift. For example, some outbreaks of the influenza virus were worse in people who had the virus before. They were able to produce massive amounts of antibodies that were not able to bind to the virus well enough. This increases the symptoms of infection without being able to fight it effectively. The influenza virus infects 3 to 5 million people per year. 250 to 500,000 of these cases lead to death. Most of these infections happen during the winter when the air is cold and the virus can more easily stay in the drier air. There have been 3 major pandemics of influenza in the past century. In 1918, an outbreak of H1N1 named the Spanish flu killed an estimated 40 to 50 million people globally. In 1957, H2N2 strain, named the Asian flu, caused the death of some 1 to 2 million people. Lastly, in 1968, H3N2, named the Hong Kong flu, is considered the least deadly of these pandemics. However, it was the cause of roughly 1 million deaths globally. While future pandemics like these are impossible to predict, scientists do agree it will happen again with a new mutation of the glycoproteins. Rapid influenza testing is key to slowing or stopping the next pandemic. Currently, polymerase chain reaction, or PCR for short, and ELISA type assays are able to accurately diagnose influenza type A and type B in 24 hours or less. These rapid tests are usually done by nasopharyngeal swabbing. Viral culture may be done to assess the exact composition of the viral envelope and identify the exact mutations on the specific influenza virus. The treatment for influenza may be accomplished if caught early. In the progression, oseltamivir, or commonly called Tamiflu, or zanamivir, also called Relenza, are common antivirals. These can be used for treatment and possibly prevention in pandemic situations. These antivirals shorten the duration of the infection by limiting the effect of the glycoprotein spike neuraminidase. Thus, viruses are not as effective at entering the mucous membranes, thereby slowing infection. Vaccination against influenza is the most effective way to control the spread of influenza by slowing infection rate and increasing herd immunity. Many different types of vaccinations exist that can protect against different strains and are made differently. Most of them are made from inactivated influenza virus grown by egg incubation. New viruses are made yearly to protect against new mutations of the glycoprotein spikes and distributed during the flu season. The effectiveness of the vaccines varies widely by year, type of influenza strain, and the age demographic, but generally they are anywhere from 40 to 80% effective. Illustrated here are my references for my research on the presentation. Please pause this video now if you'd like to review them. Picture references are listed below the picture for each slide. Thank you very much for your time and attention to my presentation on the influenza virus. Please email me with any questions or comments, and thank you for your time and have a wonderful day.